Target Malaria brings together several research institutions across Africa, North America and Europe. We are an international not-for-profit research consortium working to develop and share innovative and sustainable genetic technologies to modify mosquitoes and reduce malaria transmission. Today we will be visiting the Target Malaria insectary in Italy. The insectary is based in the Laboratory of Ecology and Genetics at Polo di Innovazione di Genomica, Genetica e Biologia, Polo GGB, in Terni. Polo GGB has been working with universities, companies and diagnostic laboratories since 2011. But this facility was purpose-built for Target Malaria in 2016.
My name is Professor Ruth Müller. I'm the Chief Manager of the Terni facilities of Polo GGB. Our facility has a large insectary, and here we generate a lot of data for Target Malaria and especially for regulatory purposes. So today we will visit our laboratories and I would be happy to show you everything.
Safety is paramount to Target Malaria. The insectary meets the international guidelines and national regulations for the containment of insects, called Arthropod Containment Level 2. Level 2 containment means that there are specific features in the lab built to ensure that insects inside the lab, in our case mosquitoes, cannot escape the insectary, and that external insects or animals cannot come in.
So here in Terni we have 3 different safety measure levels, and we have one, the infrastructure containment, and we have a lot of standard operation procedures, and we have the geographical containment. The infrastructure containment, you can see a part of this here. We are in the vestibule, which is actually a double door system, and this connects the seminar rooms and the office area from our insectary, from the contained area. Here in this vestibule, you have to change your dress, so you have to take lab coat and everybody, also the visitors. And also there's an additional safety measure. We have an air fan in front of the door to the insectary.
Traps and electric rackets are located all over the insectary to monitor and terminate any stray mosquitoes.
The third level is geographic containment. So we have African mosquitoes in the insectary, and these mosquitoes cannot survive in our Terni environment. And also we have this Anopheles species, this African species, which need a special kind of breeding sites, and this is also not around our facility.
The genetically modified mosquitoes are created in the Target Malaria insectary in London and then shipped to Terni for behavioural analysis. The main work of the lab in Terni is to analyse the behaviour of Target Malaria's genetically modified mosquitoes, to determine whether they behave like wild mosquitoes and whether they could survive in the wild. This insectary does not work with malaria parasite or infected females.
Our insectary is a very large space, which you have to imagine this way. We have a lot of wild type and genetically modified mosquitoes, and these we have in small cages but also in large cages.
The laboratory houses 4 large cages known as climatic chambers. These rooms are equipped with the latest technologies to mimic the natural ecological conditions of the mosquito species we study. The cages are built with sliding panels, so they can be resized to fit different needs. The large cages are equipped with a system that allows for the precise regulation of temperature and humidity. Light conditions are also set to mimic the daily variation at dawn and sunset. These cages are a simulation of the natural climate where malaria-carrying mosquitoes live. The cages are big enough to accommodate thousands of mosquitoes, allowing for the study of large mosquito populations in conditions as close as possible to being outside, while still being in a contained environment.
We are here in an environmental chamber with large cages, so we can even combine these cages to a supersized cage. Every cage is equipped with a swarming arena, with resting sites for the mosquitoes to recover, a sugar meal, and a place where they can lay their eggs. And Roxana is now preparing the equipment of such a cage. And we have, for example, these bricks, and this is a resting site for the mosquitoes. We wetten them with a sponge so that they can create a very, very humid microenvironment where the mosquitoes can hide during the day, rest and wait and recover a bit. Then we have also some artificial plants and some black structures where they also can use as a resting site. We have this lamp. When we put it on and put a dish with a paper filter in it, then the mosquitoes find this place and lay their eggs into this dish. Then we have these rolling structures where we put some also dishes with sugar water into it, and they can take a sugar meal. At the very end of the cage you see this square, this black square, and there the mosquito start to make a swarm. The male mosquitoes, they like this contrast of black and white, and the males swarm, and the females go into the swarm. They build a couple with one of the males and they fly out of the swarm. But this swarming is only possible when the sunset light starts. We have in the back this bit light, what you can see, and only under these conditions the mosquito like to swarm, so it would be in the evening in the nature. In general, we would like to give the mosquitoes here a very natural environment, so they can swarm, fly around, they can rest, and they have a natural light conditions, but also natural temperature variation. So in noon it's warmer than in the night, and also humidity change over the day, so we can mimic this, the daily cycle, but also the seasonal cycle of environmental parameters.
Tests and experiments are performed on the modified mosquitoes to ensure they are fit and would be able to survive in the wild. Mosquitoes go through 4 distinct life stages in their development: egg, larvae, pupae, and adult. Before they emerge as flying adults, the first 3 stages of a mosquito's life are aquatic. In our tests, we check how many days it takes the mosquito to go through these stages and their survival rate at each stage. This is done in the laboratory. Then, in the cages, we check that the mosquitoes feed, mate, and fly normally, and that they could compete with wild mosquitoes. Data collected on the modified mosquitoes in the cages is compared with the information received from the entomology teams working in Africa, who are collecting data on wild Anopheles gambiae and Anopheles colutzii behaviour. In addition to these behavioural tests, we also perform genetic tests. We mate modified mosquitoes with wild mosquitoes to check if they can produce viable offspring, and check the rate of transmission of the genetic modification in several generations.
We do integration with mosquitoes because we want to have our technology in the field, and this is important. So you have to understand, we want to transfer our genetic modification in a population which is really fit for the field. We got some mosquitoes from Burkina Faso, which are perfectly adapted to their environment. And here in Terni, we crossed them with our modified mosquitoes. First as a hybrid, and then we crossed this hybrid again and again with this wild Burkina Faso population. And the only difference between the wild type in Burkina Faso and here is that they have this modified gene. Okay, how we do it? This is a very interesting question. So we have here one cage from with the genetic modified mosquitoes. We have here one cage from the Burkina Faso, and they produce eggs. And we collect these eggs, and what we are doing then, we merge only the mothers from the one cage and the fathers from the other cage, and then they can mate and produce joint offspring.
All insectary waste is collected and burned by an external waste company. We do not burn any waste in our facility.
We collect every single waste, solid waste, in such bags, and for example the blood, the leftovers from the mosquitoes, the tissue, and then we put it in an autoclave, and we sterilize under high temperature, high pressure for around 1 hour, and then this waste can leave the contained area clean.
We hope this tour has answered some of the questions you may have around our work. At Target Malaria, we are committed to being open and accountable as we work to develop an innovative tool for malaria control